vertebrate skin appendages, such as mammalian hair, the feathers and scales of birds, and reptilian scales, all develop as genetically controlled units whose spatial self-organization involves a Turing chemical patterning system. The patterning of irregular crocodile head scales provides a fascinating exception to this paradigm. Indeed, we have previously shown that the scales covering their face and jaws do not emerge from the chemical interactions among gene products, but instead arise from a mechanical patterning process. However, until now, the nature of this process remained unknown. In our new study, we combine cutting-edge experiments, microscopy and computer simulations to solve this mystery. Initially, the embryonic crocodile jaws appear smooth. Skin folds then appear, propagate and interconnect to form irregular scales. This process generates large, elongated scales on the top of the upper jaw and smaller polygonal scales on the sides and bottom of the jaws. The mechanical patterning of crocodile head scales may be caused by compressive folding of the skin. For example, if the epidermis grows faster than the dermis, local compression causes buckling and folding, which can result in a surface pattern of polygons. To test this hypothesis, we treated Nile crocodile embryos with intravenous injections of epidermal growth factor protein, EGF, to increase epidermal growth and stiffening. Remarkably, EGF-treated embryos develop additional skin folding with more numerous and thinner elongated scales on the top of the upper jaw, and a brainy folding pattern on the sides and the bottom of the jaws. Next, we observe that EGF-treated crocodiles, hatched 4 weeks after the treatment has ended, exhibit a skin folding pattern that is partially labyrinthine and partially made of abnormally small polygonal head scales, reminiscent of those of the caiman. Therefore, variation in embryonic skin growth is a simple evolutionary mechanism that can explain the differences in head scale patterns among different species of crocodiles. Next, we build a computer simulation of crocodile head scale patterning. Using light sheet microscopy, we first reconstruct the different tissue layers found within the jaws of the crocodile embryo, namely the epidermis, the dermis, and the bone. Second, we determine the 3D architecture of the dermal collagen network and the spatial variation of skin growth. Remarkably, these mechanical simulations accurately mimic the process of head scale patterning. Starting from a smooth geometry, skin growth causes surface folds to appear, propagate and interconnect, forming irregular domains, including elongated scales on the top of the upper jaw and more regular polygonal scales on the side and bottom of the jaws. We then repeat our simulation with increased skin growth and stiffness to mimic the effect of EGF treatment. As a result, more numerous elongated scales form on the top of the jaw, and brainy folding emerges on the lateral and ventral jaw surfaces, thereby recapitulating the patterning process observed in EGF-treated crocodile embryos. Finally, we produce a theoretical morphospace of skin folding patterns, showing that variations in embryonic growth and material properties of skin layers provide a simple evolutionary mechanism for producing a vast diversity of head scale patterns observed in different crocodilian species such as those of the Nile crocodile, marsh crocodile, spectacled caiman and American alligator. In conclusion, evolution has produced 2 ways of generating crocodilian scales. As for the development of hairs and feathers, a chemical Turing system dominates the patterning of crocodile body scales. On the other hand, a mechanical system produces the crocodile head scales. Second, our experiments and computer simulations show that crocodile head scales self-organize through compressive folding that arises from the growth and mechanical properties of the skin. Third, by modifying the growth and physical properties of the skin, evolution has produced diverse head scale patterns among different crocodilian species. And finally, our analyses demonstrate that simple elastic models can efficiently recapitulate certain mechanical developmental processes,